Hi, I'm Mary Beth Iden, and we're here at the Los Angeles Zoo's Australia habitat, home to a number of species including the iconic koala. Today, I'm going to be summarizing some of the highlights of an article I wrote with One Zoo in the annual reviews of Virology. This article was about koala retrovirus, its disease dynamics, and evolution. And in some of my closing remarks, I'd like to say how this new information may have great importance for the future of the koala. Habitat loss and diseases such as malignant neoplasias currently threaten the koala. A retrovirus has long been suspected as the agent responsible for malignant neoplasias in koalas. In 2000, a retrovirus called CORV for koala retrovirus was isolated from koalas in its integrated form within the koala genome by a team of Australian scientists. If CORV is the cause of koala malignancies, it would have to be passed from one koala to another as an infectious virus, rather than by genetic inheritance as an endogenous one. However, Australian scientists, using several lines of scientific evidence, showed that CORV was in fact an endogenous virus, inherited from the parental genome. Yet puzzlingly, southern blot analysis revealed a second pattern of viral integration, suggesting new copies of the virus were being inserted non-genetically in an active infection in the koala population as a whole. It took historicogenetic research into the koala genome using koala museum specimens and work done here at the Los Angeles Zoo with well-pedigreed koala families to show that there was in fact two species of the koala retrovirus. One, called CORV-A, endogenized in the koala genome more than a century ago and is stably genetically integrated and is most likely non-pathogenic. A second, which we called CORV-B, and which may have arisen from CORV-A itself, is transmitted horizontally by infection rather than inheritance. CORV-B, or a closely related virus, is the most likely cause of lymphomas in koalas. How did we use the koala breeding programs at the Los Angeles Zoo and the San Diego Zoo to zero in on CORV-B? Well, the San Diego Zoo, then under the direction of Dr. Jeff Pye, has maintained the largest colony of koalas outside of Australia. These koalas have been maintained with limited outbreeding for more than 30 years and have a very low incidence of lymphoma in the 21st century. The Los Angeles Zoo, under the direction of Dr. Cindy Stadler, has continuously assimilated captive Australian koalas into its breeding program, and these koalas have a relatively high incidence of lymphomas. However, we were also able to isolate a second koala retrovirus, which we called CORV-B, from some of the koalas at the Los Angeles Zoo, but from none of the koalas at the San Diego Zoo. The pattern of CORV-B infection and lymphoma prevalent in one Los Angeles Zoo breeding family in particular allowed the role of CORV-B in disease to emerge. Specifically, what we did is we examined the 15 descendants of the Los Angeles koalas Dodger and Jane. Now, both Jane and Dodger were CORV-A positive, but Jane was also CORV-B positive. However, only CORV-B positive female descendants transmitted virus to their offspring, strongly suggesting that the virus is transmitted as an infectious particle, whether in utero before the joey migrates to the pouch or in the dam's milk. All the koalas that died of lymphomas at the LA Zoo were CORV-B positive, suggesting a strong association between the presence of CORV-B and pathogenic outcomes, and that CORV-B is the cause of koala lymphoma. How should we use this information about CORV endogenization, transmission, and pathogenesis? First, koala breeding programs cannot be sustained with koalas that are totally retrovirus-free. There are just too few CORV-free koalas in the world to do this. However, it is now clear that there are multiple CORVs with different modes of transmission and pathogenesis, and we can at least ensure that the koalas bred in zoos are free of the most pathogenic strains of CORV. We can also streamline real-time CORV identification in Australia koalas to guide viral eradication programs using both antiretroviral drug treatment and eventually vaccination. Ultimately, understanding the history of CORVs and their impact on worldwide koala populations will help us understand retroviral evolution throughout the planet's mammalian populations, including our own species.